The forest is dying in slow motion. You can hear it if you listen, not with drama, not with collapse, but with a kind of quiet subtraction. The wind moves through what used to be canopy. Now it moves through absence, through grey skeletal fingers reaching upward, through bark that peels like old wallpaper from a house that has already been abandoned. This is an ash woodland in the English Midlands, or it was. Fraxinus excelsior, the common ash, has stood in these islands for 10,000 years. It arrived after the glaciers retreated, walking northward at the pace of seeds and centuries, threading itself into the mythology of Norse cosmology, into the hedgerows of medieval England, into the lungs of songbirds and the shelter of foxes and the green cathedral ceiling of 10,000 country lanes. For 10 millennia, this tree held its place, quiet, enduring, unremarkable in the way that only truly essential things can be. And then, sometime around 2012, the dying began. Not suddenly, not with fire or storm, with a fungus so small you cannot see it without a lens. Hymenopsyphus fraxineus, a pathogen that originated in Asia, where native ash species had evolved alongside it for millions of years, building tolerance across geological time. But when it arrived in Europe, probably on nursery stock, probably on a plant somebody imported without knowing what was traveling with it, it found a continent of ash trees that had never encountered it, had no memory of it, had no defense. The disease it causes is called Ash Dieback, and it is, by any measure, one of the most devastating tree diseases in recorded history. Within a decade, more than 1 billion ash trees across Europe were infected. In the United Kingdom alone, the cost projections were staggering. Some estimates placed the total economic damage at £15 billion. Others said 12. The numbers shifted, but the trajectory did not. This was going to be a catastrophe. The question was not whether the ash would be lost, but how completely. Governments responded, scientists scrambled, research programs were funded, breeding initiatives were launched. The logic was urgent and simple: find the survivors. Somewhere in the hundreds of millions of infected trees, there had to be individuals that were resisting, trees with something in their genetic architecture that the fungus could not overcome. Find them, cross-pollinate them, grow their offspring and replant a resilient forest. This is what we call a selective breeding program. It is the same principle that gave us disease-resistant potato varieties, drought-tolerant wheat, apples that don't brown. We have been doing this with crops for 10,000 years. Surely we could do it with a tree. The project was called the Ash Archive. Scientists from across the UK collected seeds and cuttings from thousands of ash trees, from ancient woodland, from hedgerows, from isolated specimens in churchyards and field margins. They grew them in protected nurseries. They exposed them to the pathogen under controlled conditions and watched and waited, and what they found was real. Around 5% of ash trees showed measurable tolerance to the fungus. Not immunity, tolerance. The disease still colonized them, but slowly. The crown dieback that killed other trees within 2 or 3 seasons was, in these individuals, reduced. They limped forward, they survived. 5% sounds small, but 5% of the ash trees in Britain alone is still millions of individuals. If those trees could be identified, cultivated and used as the genetic foundation for a new ash woodland, the species could be saved. Or so the logic went. And here is where the story becomes something more complicated than science. The breeding program moved forward, it continues today, but something was becoming apparent in the data, in the field surveys, in the quiet accumulation of observation from ecologists working across Britain and Ireland and Scandinavia. The numbers were not adding up the way the models had predicted. The 5% tolerant trees were surviving, yes, but the ash woodland wasn't recovering, even where infected trees had been removed. Even where tolerant specimens had been replanted, the ecosystem beneath them was not returning. The fungi on the forest floor, the mycorrhizal networks, the decomposer communities, had shifted in ways nobody had fully anticipated. The light regime had changed. The invertebrate populations that depended on ash leaf litter had declined. The birds that depended on those invertebrates had declined. The predators of those birds had declined. You cannot save a tree by saving only the tree. This sounds obvious when you say it out loud, but the history of conservation is littered with examples of exactly this mistake, of treating a species as a unit to be preserved, rather than a relationship to be understood. We saved the red kite and found its recovery incomplete until its prey base recovered too. We saved the white-tailed eagle and discovered it needed not just nesting sites, but healthy fish populations and undisturbed coastlines, and the absence of lead ammunition in carcasses. Every species is the sum of its connections. The ash tree, it turns out, supports more than 300 species of wildlife, insects, lichens, birds, bats, mammals, in relationships that have been woven together across millennia. You cannot simply replant the tree and expect the web to reassemble. The web has its own memory, its own logic, its own pace. And so by around 2018, the mood in the scientific community had shifted from urgent optimism to something quieter, more uncertain. The rescue plan was real and necessary and was producing real and necessary results. But it was not by itself going to be enough. And then, and this is the part of the story that changes everything, somebody looked more carefully at the trees that were dying, not the survivors, the dead ones, the ones nobody had been paying attention to because they had already been written off. A team of researchers, working across field sites in Ireland and Wales, began examining the remains of ash trees that had succumbed to dieback. They were looking initially at the fungal communities present in the dead wood, a fairly standard ecological survey, the kind of work that generates data sets rather than headlines. But one of the graduate students on the project, a young woman named Dr. Aoife Nídhuibéin, working out of University College Dublin, noticed something in the microbiome data that did not fit the expected pattern. In the bark tissue of some dead and dying ash trees, she was finding fungal DNA sequences that did not belong to Hymenopsyphus fraxineus. They belonged to something else, something closely related, something native. She flagged it. Her supervisor told her it was probably contamination. She ran the samples again. The signal remained. She published a preliminary note. It was largely ignored, but she kept looking. What she eventually documented, and what was confirmed by subsequent independent analysis, was that certain ash trees, as they died, were fostering the growth of a native fungal endophyte, an organism that lives inside plant tissue without causing disease. And this endophyte, in laboratory conditions, showed direct antagonistic activity against Hymenopsyphus fraxineus. It produced compounds that inhibited the pathogen's growth. It competed for the same biochemical space. In other words, inside the dying trees, a defense was emerging, not in the trees themselves, not in their genetics, but in their microbiome, in the invisible community of organisms that had evolved with them across the same 10,000 years, and that were now, in response to a novel threat, mounting something that looked almost like an immune response. Almost. We have to be careful here. Science does not traffic in poetry, and the temptation to narrate this as a triumphant last stand is the kind of temptation that has led popular science astray before. The endophyte does not save the trees still die. But what the endophyte appears to do, in conditions that researchers are still working to understand, is slow the progression of the disease, create pockets of reduced infection pressure, and in some cases allow neighboring trees, including the 5% of genetically tolerant individuals, more time, more seasons, more opportunity to reproduce. It is not salvation, it is something more interesting than salvation. It is a system responding to itself. The broader significance of this discovery extends well beyond the ash. It points toward a principle that ecologists have been circling for decades, but which has been difficult to communicate to a public and to a policy world that prefers clean solutions to complex ones. The principle is this: ecosystems do not need to be saved, they need to be listened to. The ash woodland was not a passive victim awaiting human intervention. Even as it died, even as the grey skeletons accumulated across the hillsides of England and Wales and Ireland, processes were unfolding at scales too small to see without instruments and too slow to notice without patience. The system was adapting, not fast enough to save itself alone. The pathogen had arrived too suddenly, too virulently for that. But it was not passive, it was working. What humans can do, what we must do, is create the conditions for those processes to continue. To give the system time, to resist the impulse to tear out the dead trees, to replant monocultures of selected specimens, to impose our own narrative of what recovery should look like onto a forest that has its own ideas about the matter. This is the philosophy of rewilding, and it is often misunderstood. It is not abandonment. It is not neglect. It is the disciplined and humble act of stepping back, of acknowledging that the intelligence distributed across millions of organisms, refined across millions of years, is likely wiser about the specific problem of survival in this specific place than any research program, however well-funded and however well-intentioned. The Ash archive continues. The selective breeding work continues. Those 5% of tolerant trees matter enormously, and the science of understanding their genetic architecture, which genes confer tolerance, and whether those genes can be broadened through careful breeding, is work that will take decades and that deserves every pound and every researcher hour invested in it. But alongside that work, something else is being tried. Across a handful of sites in the UK and Ireland, ecologists are experimenting with what they call assisted natural regeneration, the practice of reducing intervention, allowing dead wood to remain, preventing the grazing that would otherwise suppress new ash seedlings, and watching what the forest does with the time it is given. Early results, as of the most recent surveys, are quietly remarkable. In sites where dead ash trees have been left standing, where the logs have been allowed to rot, where the fungal communities have been allowed to cycle the nutrients back into the soil, the seedling populations of ash contain a higher proportion of tolerant genotypes than the surrounding woodland. This is natural selection happening in real time. The pathogen itself is acting as a filter, allowing only the resistant individuals to reach adulthood, which means the woodland is slowly, painfully slowly, selecting for its own survival. And in those seedlings, researchers have begun finding the same endophyte communities that Awif Nidobain first documented in the dead trees. The microbiome is transmitting, not through genetics, not through deliberate human action, but through the soil and air and bark contact and whatever invisible conversations organisms hold in the language of chemistry and proximity. The knowledge is passing on. There is something almost unbearably moving about this. Not in a sentimental sense, not in the way we sometimes dress nature in human emotion because it makes us feel connected to something larger than ourselves, but in a genuinely empirical sense. When you understand what is happening, when you understand that 10,000 years of co-evolution are producing, even now, even in crisis, even in loss, responses that no laboratory could have engineered, you are confronted with a scale of intelligence that rearranges something in how you think about the world. We live on a planet that has been solving problems for 4 billion years. Problems of survival, of disease, of climate, of catastrophe. It has survived mass extinctions. It has survived asteroid impacts. It has remade itself after volcanic epochs that blotted out the sun for 1,000 years. It does not do this cleanly or painlessly or without tremendous loss, but it does it. The Ash Dieback crisis is a catastrophe. We should not minimize that. The loss of these trees from the landscape is a loss that compounds across every species connected to them, across the aesthetics of the countryside, across the cultural memory embedded in hedgerows and ancient woodland. It is a loss that humans caused through the casual transportation of plant material across ecological boundaries that evolution had spent millions of years maintaining. That responsibility is real, and it should not be softened. But what the endophyte discovery tells us, what the natural regeneration data is beginning to suggest, is that the story is not over, that it was never simply a story of loss, that inside the dying, something was always also beginning. The ash trees standing dead in the Welsh hillsides right now, their bark grey, their crowns bare against a winter sky, are not monuments to failure. They are infrastructure. They are habitat for the woodpeckers and the bark beetles and the hoverflies and the hole-nesting birds. They are rotting into the soil, feeding the mycorrhizal networks that the next generation of trees will depend on. They are, in their dying, still giving. Maybe that is what we have always misunderstood about forests. We think of them as things that stand, as structures defined by height and canopy and living green. But a forest is also what falls, what rots, what becomes soil, what persists in the chemistry of the earth long after the last visible trace of the tree has gone. The ash is not dying, it is becoming something else. And somewhere, in a quiet field in County Clare, or on a Welsh hillside where the light falls thin and golden through bare branches in late afternoon, a seedling is pushing up through leaf litter. It is 2 inches tall, it has 3 leaves. It carries in its cells the tolerance of its parent, and in the soil around its roots the same fungal companion that helped that parent survive. It doesn't know it's a rescue, it just grows.